Remember how we talked about the evolutionary perspective of zoology, how taking a look at organisms' relationships to other organisms indicate how closely related they are and therefore how they evolved from each other. Well, taxonomy is a big piece of that. When we figure those pieces out, we can classify organisms. We can put them into groups based on things that they have in common. So this presentation is going to be about how we classify organisms, and the study of classification is taxonomy. Linnaeus is considered the father of taxonomy. He was the first person to start organizing organisms. And he started by just identifying plants and animals, and everything had to fall into one of those two categories. We now know there's a lot more diversity of life, but at the time, in the 1700s, he didn't know any better. So there were plants and there were animals, and he came up with a way of naming those organisms. It's a binomial nomenclature, so there's two words. Each species has two words that are Latin. One is the genus and one is the species. For example, your house cat is Felis domesticus. The genus is a noun, the species is an adjective. You'll notice that it's written in italics, so you can see that here. You can either write it in italics or you can underline it. Also notice that the first word, the genus, is capitalized, and the second word, the species, is not capitalized. And so this is the format that we've kept all the way from the 1700s to now on how we name organisms. So back in the 1700s, when Linnaeus came up with this binomial nomenclature, he decided it should be in Latin. And that was because Latin is a language that was unchanging. It was also a language that was studied by all educated people at the time. So those that were currently doing work on studying plants and studying animals would be able to name those plants and animals because they would know Latin. Linnaeus identified kingdom, phylum, class, order, family, genus, species. I'm sure you've heard that in biology. That system was used to narrow and classify organisms, kingdom being the most broad and species being the most specific. Each one of those levels is considered a taxa. We later added on domain because we realized, wow, bacteria, whew, there's so much of it. So we added that extra layer, but ultimately his system from the 1700s is still in place. We've made changes because originally fungus was identified as a plant, so that now has its own kingdom. But this general system has stayed in place since the 1700s, which is pretty amazing. Today we use a 3-domain system, and within those 3 domains there are 6 kingdoms. One of the domains is bacteria, and all of the bacteria domain falls into the kingdom of bacteria. Then there's the archaebacteria, that's another domain, and all of the archaebacteria falls into the kingdom archaea. Now, it's important to understand that both of those are prokaryotic. All bacteria are prokaryotic cells. Then we have the last of the 3 domains, eukaryota, and those are all the eukaryotic cells. So that's your plants, that's your animals, your fungus, and your protists. So within the bacteria domain, or the eubacteria, you'll see it both ways, you have common bacteria. These are going to be the ones that get you sick, that they're everywhere. They're currently in your mouth, they're in your intestines. It's E. coli, it's stuff that's everywhere, that lives in normal conditions. The second domain is the archaebacteria, and these are bacteria that are crazy. They can live in extreme environments. They can live in extreme temperatures, extreme salt conditions, extreme acid, extreme base conditions. These are the bacteria you see growing in the hot springs. These are bacteria you're not going to see everywhere. They have a very limited place where they can live because they can tolerate extreme events. And then finally we have the eukaryotes. And again, that's what we typically think of when we think of living things. We think of animals, we think of plants. There's also fungi as well as protists. And protists are very simple organisms. They're still single-celled. And that's how we go from single cell to these guys, which are multicellular, although some fungi are single cellular. So let's take a look at an example of the classification of an organism, and we're going to use humans as that example. Our domain is Eukaryota. We are animals. We fall in the phylum of chordates. Our class is mammal. Our order is primate. Our family is hominid. We are in the genus Homo and the species sapien, and therefore our scientific name is Homo sapiens. And you can see it's written in proper scientific notation. One of the ways that we started to organize the different organisms that were found was based on body symmetry. And body symmetry is how the parts of the animal are arranged around a point or an axis. We can initially determine how evolved an organism is based on the symmetry of the organism. We humans have bilateral symmetry. Our left side looks just like our right side. It's a mirror image of it. That bilateral symmetry is actually considered fairly evolved. So anything that doesn't have bilateral symmetry typically is less evolved. And we'll see how that plays out in a second. So some of the most primitive animals completely lack symmetry. They're asymmetrical. There's an absence of a central point or an axis around which the body parts are arranged. And so sponges, as you can see from this picture, are asymmetrical. At no point can we find an axis that we could put a mirror and the one side is the same as the other. As a result, sponges are also one of the most primitive animals that exist. And so as we start studying the different phyla of animals, we'll start with sponges. Another type of symmetry is radial symmetry. This is the arrangement of body parts around a central axis. So here we have starfish, and you can see there's that central axis, and it's arranged around that central axis. Now keep in mind that this radial symmetry in starfish we find in the phylum Echinoderms, and they're kind of a weird special case in evolution where they branched off at a different time. Another type of radial symmetry we see in sea anemone, and sea anemone are cnidarians, which we'll study shortly after we study sponges. And so their mouth, this central point, is where they all start to radiate out from. And so you can see they have that radial symmetry, making them more advanced than a sponge that doesn't have any symmetry, but not quite at the point where it has bilateral symmetry, so less evolved than humans. Finally, we get to bilateral symmetry, and this is the arrangement of body parts, that there's a single plane that divides the animal into left and right mirror images. So my left eye and my right eye are the same size. My left ear and right ear are placed on the same spot on my skull, just on different sides of it. And so if we use this median plane here, that's going to divide our body in that left and right side, and that's going to be a mirror image. And typically bilateral symmetry is indicative of the most evolved organisms. And most of the organisms that you think of, most of the animals you think of, are typically going to have bilateral symmetry, because most of the time when we think of animals, we think of primates, we think of chordates, so they're all more advanced. Here you can see a side-by-side comparison. We start over here on the right with the sponge, which has no symmetry. It's the asymmetrical version. Then we go on to the radial symmetry that we saw. This is a coral, but they're closely related to the sea anemone. And finally, a beetle, an insect, which is part of the insects, which are fairly well evolved, and it has bilateral symmetry. So you can see how they compare. Another way that we can determine how evolved an organism is based on the organization of its cellular structure. For instance, unicellular or cellular aggregates are going to be less evolved because they're single celled. Once we start getting multicellular organisms, that's a sign of evolution having taken place. We move from less evolved single cells to more intricate multicellular organisms. One of the first things that we're going to see is diploblastic, and that's 2 tissue layers. And you're going to see this when we study sponges and when we study cnidarians with jellyfish and things like that. They're going to have 2 cell layers. They have the ectoderm, which we talked about, that's that outer layer, and then they're going to have the endoderm. Notice that they don't have that mesoderm. They have the gut inside, and then in between, instead of having the mesoderm, they have a mesoglia, and that mesoglia is non-cellular. It's the space between them, but there are no cells in there, and that's kind of what makes like jellyfish squishy. It's that mesoglia. It's not living, but it's still in between the endoderm and the ectoderm. Moving up that evolutionary chain, we have the triploblastic. That's 3 cell layers. That's where we finally get that mesoderm. So we have the ectoderm on the outside. We have the endoderm on the inside, with creating the tube for the gut. And then in between, we have the mesoderm, which is now cellular instead of non-cellular. These organisms typically have bilateral symmetry, and they're very active. By the time that mesoderm forms, remember what gets formed from mesoderm are things like blood and kidneys and liver. And so when that mesoderm's present, there's typically body organs that go along with it. Within that triploblastic organization, there are 3 different patterns that we'll see. And what's really kind of cool is we will study how these evolve as we look at worms. So one of the things we're going to start with is flatworms, and they're the least evolved, and they're asolemates, which means that their mesoderm forms a relatively solid mass. They don't have many organs and things like that. The mesoderm's not used to make body organs and body cavities. Then we'll move up to roundworms, and they're pseudosolemates, and that body cavity, there's a body cavity that lines the mesoderm, but it's not a true body cavity. It's kind of static. And we'll talk more about this when we get to worms. And then finally, we have solemates, and they're the ones that have the true, here's the ectoderm, here's the endoderm, and finally you have that mesoderm forming meaningful organs and organ systems. And we'll see solemates come up with segmented worms. So when we study worms, we'll see this progression, and we'll review this idea, but I want you to see where it's coming from now, because it ties into that taxonomy and how things evolve. So we'll come back to this idea later. So when you come across an organism, let's say you find an insect when you're outside and you have no idea what it is. Typically, the way that we figure out what organism it is by using a dichotomous key. And that prefix di means 2. So we're going to use 2 different choices that allows us to identify what that animal is. And there are books about this for animals, for plants, for insects, which I know are animals, but they get more specific. And you can see there's always 2 choices. For instance, are there feathers? Yes is one way, no is another way. If the answer to feathers is yes, does it swim? Yes or no? This is one of the ways that we can determine an unknown organism. So before we get all excited, like, oh, I just found this bug and it's brand new, no one ever found it before, it goes through multiple different dichotomous keys to make sure, did we really discover something new, or is it just a variation of a pre-existing, already found, already named bug? So what we're going to do now is you're going to spend some time playing with dichotomous keys and making dichotomous keys to reinforce this idea.